Hey guys, okay, so this is day 2 of our biotechnology notes. I'm going to skip through all of the stuff that we did yesterday really quick, and we are going to start with PCR. There's this really good 3-minute video, so if you're still confused after this, you can watch that. It might help you understand a little bit better. But PCR is polymerase chain reaction. This is going to use polymerases to copy DNA segments, and we know polymerase is an enzyme because it ends in our suffix ase. PCR makes many copies of a specific DNA sequence in just a few hours. So basically, you're going to have this DNA fragment, and we're trying to make more copies of it. And there's several reasons that we do that. We'll talk about those in a minute. And so PCR is going to make many copies of this pretty quickly. So PCR amplifies DNA samples, just means to make a large number. So amplify, just like you amplify sound. Same thing here. PCR is similar to DNA replication, which is happening inside your body right now, inside our cells, getting ready for mitosis. So same thing, we're just doing it like with technology. So we're doing it outside of our bodies, outside of the natural place. PCR is a 3-step process, and it uses 4 materials. The first material is DNA to be copied, so you've got to have that fragment that you're wanting to make more of. You have to have DNA polymerase to be able to put on those nucleotides, just like in DNA replication when we learned about that a little time back. We talked about polymerase being the thing that is putting on those bases, so that's going to do that for PCR. We have to have A, T, C, and G nucleotides. Those are our bases, right? And so we have to have those nucleotides available that when that's what's called for to make the copy of our original DNA fragment, we're going to have those available so that polymerase can put those on. And then we're going to have primers, and that's going to be where that starts. So you can see here DNA strands, our free nucleotides, all of that. So each PCR cycle doubles the number of DNA molecules. So we look at our picture here. We have 1, then we have 2, then we have 4, then we have 8. After 30 cycles, more than 1 billion copies have been made. So that is a lot of copies of this one DNA fragment, which is really cool. So a DNA fingerprint is a type of restriction map. DNA fingerprints are based on parts of an individual's DNA that can be used for identification. So it's based on non-coding regions of our DNA. So these are going to be the regions that are not coding for those proteins. And they're going to have repeating DNA sequences. So ACGT, 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 or AATT, AATT, whatever it is, it's repeating. The number of repeats differs between people. So that's your DNA we know is different. Otherwise, we'd be the same person. So these repeats are going to look different between everybody. And the banding pattern on a gel is a DNA fingerprint. So that banding pattern that we talked about with gel electrophoresis yesterday, that is going to be a DNA fingerprint. And there you have it on the right. You can see the little DNA fingerprint or the banding patterns. So it's used for identification. DNA fingerprinting depends on the probability of a match. Many people have the same number of repeats in a certain region of DNA, but unique patterns of repeats are identified using PCR and electrophoresis. So there are some times that you have the same area that looks the same between people, but the probability that 2 people share identical numbers of repeats in several locations is very small. So maybe in one location you have the same number of repeats, but for you to have the same number of repeats between a lot of locations is pretty low. So you can see some banding patterns there. You see the mother, child 1, child 2, and the father, and you can see how child 1 and the mother have a similar pattern up there at the top, and then how child 2 and the father have a similar banding pattern at the top. So individual probabilities are multiplied to find the overall probability of 2 DNA fingerprints randomly matching. So if we looked at one location and there's a 1 in 500 chance that that matches another person, and then another location it's a 1 in 90, and another location it's a 1 in 120. So when we multiply all those together, there's a 1 in 5.4 million chance that those 2 people are randomly the same in all of those locations. So several regions of DNA are used to make DNA fingerprints, and 13 to 20 matching regions are required for a legal DNA match between DNA evidence and a suspect's DNA sample. So not just 3 like we have up here at this math problem here, but it's 13 to 20 of those have to be multiplied to match, and they have to look exactly the same for a suspect's DNA to be perfectly matched with a crime scene DNA. DNA fingerprinting is used in several ways. Evidence in criminal cases, paternity tests, immigration requests, studying biodiversity, and tracking genetically modified crops. So that's all we're going to do for today. As always, let me know if anything didn't make sense. Happy day 2 of biotechnology and day 7 of quarantine, or day 12. I don't know what day it is, but it's been quite a few. So see you guys later.